So what I would like to give in the next 40 minutes is an overview of a work that we have been doing in the last few years. But the results that I will present is mostly in the last 2 years, trying to understand. First, the motivation was to understand the source of coherent splitting in claiming these experiments in photosynthetic systems at room temperature. But it became more in understanding really how these systems work and what are the underlying principles of energy transfer in this light harvesting antenna. On one side, try to understand whether really you can have truly signatures of no classical behavior there, whether you can enhance those sources of no classicality, maybe in artificial systems, whether this might play any role actually in the biology. And also the motivation is the physics behind these systems is pretty interesting. Yeah? So independently of whether it works or no for biology, I personally find that the physics behind is really fascinating. So that's my main motivation to study these systems. So in my understanding, after a few years of working in this field, I believe that there are 3 questions that people have tried to understand. The color that shows here is not very good for the room with the light. It doesn't really matter, but I'll say here. So the first question is we would like to understand, in the quest for the quantum in these systems, we would like to understand how you can describe, characterize, and quantify that non-classical quantum behavior. So one big question is whether there are specific measures that we can use to actually quantify this non-classical when I say quantum, I mean exactly not classical, not just a quantum analogous of a classical system. So we would like to understand how we can describe and quantify this quantum character, whether of the dynamics of the system or in general of the properties of the system. The second point is whether, if there is this non-classical behavior, is to understand what is the source of that non-classicality, and whether, if we understand what that source is, then whether we can manipulate that one. And finally, of course, given that these are biomolecules that are embedded in a living organism, the question is whether what we are studying here in molecules isolated from the living organism will play any role actually in the biology of the organism. That's a much harder question. So in the next 35 minutes, what I would like to show is a few insights into answering these questions, at least from the perspective of the work that we've done. So first, in the first part of the talk, I would like to describe how, for the excitation dynamics of these biomolecules, there is a fundamental role for the vibrations that are involved in the transport. In particular, I would like to show that when you consider into account a more detailed structure of the vibrations that are involved, you can actually identify basic functional units in these biomolecules. And these small functional units in these biomolecules have a physics that is rich and that can give you some answers into what coherence might be doing in these systems. Then I would like to pose from this what could be a possible role really for these coherences from these studies. And finally, given that we are focusing here on the vibrations that are participating in the transfer, I would like to show an example of how, depending on the energy scale of the vibrations that are involved, it is possible to truly identify non-classical features during the dynamics of these molecules at room temperature. In the end, I will try to discuss what experimental techniques might be possible to distinguish them, given that the techniques that are developed so far are mostly aimed at observables of the electronic degrees of freedom rather than the vibrational degrees of freedom. So as I said, the first part, I would like to focus on this part in describing that there is a fundamental role for some vibrations that are involved in the transport. In particular, the vibrations that I focus are these ones that are in close resonance, not perfectly matching, but in close resonance with excitonic transitions in the system. This is a work that, well, I made a mistake here. This is a work that has been done not by Edward, who is a PhD student working with me, but actually was a postdoc a former postdoc, Avinash Kohli. It's a working collaboration with Greg Scholes, and the second author is Edward, but I made a mistake in writing here. And what brought me into understand the role of these vibrational modes in these systems was try to understand deeply what was happening in this cryptophyte light harvesting antenna. Those are, if you remember from Elisabeta's talk, those are the antennas that Elisabeta is studying. The first reports on coherence between in these systems, which means oscillations of what could be possible quantum superpositions of excitonic states in the system, were reported by the group of Greg Scholes, where Elisabeta was working. And what is really fascinating in this system is that different from other antennae, like for instance LH2 in purple bacteria, the system that Ian was mentioning or the Mohan mentioned at the beginning of the conference, the distances between the chromophores in these systems is very large. So on average, it's about 20 Armstrong. This is very large in comparison to LH2, when the average between pigment is less than 10 Armstrongs. So then, if the design principle of this system is to use dipole-dipole interactions to transfer energy, then these distances are not very favorable towards that transfer of energy. So the question is, what is then helping this system still to be pretty efficient in transfer the energy, because there is no difference in the energy of transfer here overall in comparison to other antennae. So in trying to answer this question, here this picture is just showing the absorption. So this is a cryptophyte algae that absorbs at around 545 nanometers. That's where the name comes from. And the calculations that I'm showing here, the black curve is the experimental data, and these other two curves here are calculations that we've done with different theories. So as you can see, we reproduce okay in this part, but there is something going on here. So I will try to give an insight of what is going on. Why do we have this difference? In principle, you would say, oh, well, it's not so bad because you are actually capturing the broadening of the absorption. But actually, I will speculate at the end that the phenomena, this coupling to these vibrations, are actually coming into explaining this absorption that simple theories that do not take them into account cannot reproduce. So when it comes to the transfer of energy, it is true that the arrangement of the chromophores, that means the dipole-dipole interaction, does determine a lot how this energy is going to transfer. But probably at the same level of fundamental aspect for this energy transfer is the interaction of these electronic degrees of freedom with what we call the vibrational environment that they have, or in general, the phonon environment that they might encounter. So the spectral density is the quantity that normally captures this interaction. What I'm showing here is a spectral density that has been determined indirectly. That means that after linear spectroscopy, having results of linear spectroscopy, you look for a model of system-environment interaction that is able to reproduce your linear spectra. So this is not a directly measurement of the spectral density, but it's an indirect measurement. It's a good place to start. And this spectral density captures two things. So if you see in the lower energy range, between 0 and 400 wave numbers, the KT is around 200 wave numbers. You see that the spectral density is described by a continuous distribution. This means that you have a broad, kind of like a bulk modes that are propagating. And these modes propagating there are characterized by this particular cutoff frequency that is around this, where it peaks. It means around energies of that mode, the coupling of your electronic system to those energies are larger. But you also see that the system that I'm considering has 7 pigments. So you can imagine, you can do the account, and you can imagine how many excitonic transitions that you are going to have. So it's many excitonic transitions. Only a few of them are within the scale of this coupling that I have for this continuous mode. This continuous distribution, in the case of these biomolecules, can come from the solvent in which the molecules are involved, but also from bulk modes of the protein to which these chromophores are attached. But the most striking feature of this interaction with this phonon environment are these peaks that you see in this spectral density, and how many of these excitonic transitions that are represented by these blue lines match some of these peaks. These peaks are representing a strong interaction between your electronic degrees of freedom and specific vibrational modes that are localized in each molecule. In this case, for instance, you can see there is this peak that is around 1,000. This is in the specific value here is 11 wave numbers. And there is a quite strong interaction. So the strain of this peak is larger than in this case. But you also see that there are other peaks around 700, which also have many transitions matching with that peak. In this case, we speculate, because I don't have exactly the Raman spectroscopy of this molecule, but previous Raman studies indicates that these peaks correspond to intramolecular vibrations. So it could be a stretching mode, it could be a CO mode. But what is important here is that it's an intramolecular vibration. In other cases, because there are other systems, these localized modes, even though they are local, they might come from intermolecular interactions. So it could be a collective mode of the protein and the chromophore. It's still local, but it does not come from internal vibration. In this case, however, it seems that these particular couplings to these strong modes, these are modes coming from the chromophores, which is not surprising. The billings that we have here are very different to other billings, chemically very different to other billings. Yeah?
Excuse me, the vibrational spectrum is, comes from your computation, from your model, or?
No, they're
both experimental.
This is not a vibrational spectrum. This is a spectral density, which is different. It's the phonon density weighted by the couple to the electron degrees of freedom. So, of course, they are related, but this is a weighting. So if you only have the vibrational spectrum, you would have many, many peaks, probably many of them are the same. So these intensities would be different, because this one, in this case, they are weighted by that. So it doesn't come from an ab initio calculation. It comes from, you have your linear spectra, absorption and fluorescence, transient and function, transient and fluorescence, and then you propose a model for this spectral density that reproduces to a very good approximation your results. This is all within a model. So this is one of the key, I would say, challenges in this field. It's actually obtaining an accurate measurement of this spectral density. That's why during, there is not the person who was describing this disentanglement between electron and phonon in the lab. That's what I was interested in that, because maybe a technique like that might give some insight into this, given that the transport is so sensitive to the structure of these interactions. But to begin with, it's a very good approximation, and it gives us a very good insight of what is happening here. So as you see, many of these transitions are actually matching these vibrational components, while only a few of them are around the low energy distribution. What this low energy background does is that, given that it is around the thermal energy scale, this background will induce transitions between states that are close by in energy. In contrast, and while doing so, they decohere any superposition that you might have. In contrast, these localized vibrations, these sharp peaks here, are representing a Capleton specific. Let's begin with a classical picture, classical oscillator there. And that means that the oscillator will give a bank of a particular energy to induce the transfer. So these two backgrounds then do very different things. While one is inducing transitions between excitons that are nearby in energy, the other one is able to induce transfer between excitons that have very well far apart energies. And this is the key for the efficiency of transfer in these systems. So I'm going to go back for a moment to this one. As I mentioned at the beginning, the distances between these chromophores are very large. One thing that I didn't mention is that the on-site energies, that means the energy of the excited state of each of these molecules, they all have very different on-site energies. So there is a large energy gap to fill between each of these chromophores. Therefore, what these high energy modes are doing is essentially bridging that gap. So not only bridge the gap in energy, but also in distance. And that's what makes these systems very efficient, or not very efficient, but to do what they need to do. So from that analysis, just by this analysis, you can see that then there is a fundamental role for this coupling to these particular vibrational modes that in this case are localized. So having this in mind, then we set up to the task to understand what these modes were doing. And we have this paper that is currently is going to be published in JCP. You can have a previous version of the paper in the archive. I haven't downloaded the new version, but I can send it to you if you are interested. And essentially, we show in that paper several and key roles of these quantized vibrations for the system. The setup we use in this case is a setup that is able to capture a strong coupling to the bath. It's a setup that is accurate in the case that you have these large energy gap differences, and it's a setup that is good when you have a small coupling in comparison to that. The setup is based on a polaron transformation in which you assume that your excitation, when it is in interaction with the bath, gets dressed by this phonon bath, and then you now have an effective interaction between these dressed states. Then doing a transformation back after you calculate your dynamics, then you can have your dynamics in the original lab framework, and then you are able to compute dynamics. For details, you can come and see this. But for this talk, what I wanted to show was the physics that is behind this. So this system is divided in 4 subunits. The division of the subunits comes from the background of these chromophores. You have different protein subunits which are attached in these chromophores. What these high energy modes are doing is they are bridging the distances between these subunits in such a way that if you have an excitation that has been absorbed with the higher chromophores, as soon as that happens, then energy is distributed across the whole antenna. If the modes were not there, what would happen is that you will have transfer between nearby energy states. So then you will have excitation that, for instance, it started here, and then maybe slowly goes here, and then here. But you will not have that broad distribution, which is what I'm capturing here. So you already see, without any quantum coherent dynamics necessarily, you can already see that these high energy modes are fundamental for the biological function of this, because this antenna need to transfer to other antenna. And for that to happen, excitation needs to be distributed across the whole antenna. Now, this is a result that you don't need any fancy theory to be predicted, because you can predict already this with Foster theory, precisely because you have large energy gaps and small electronic interactions. However, you do need to go to a more fancy description of the dynamics if you want to understand the coherences and the beating of that coherences that have been observed in the experiments. And that is this kind of fancy. It's essentially an extension of this Foster theory that accounts for that dynamics. So within this framework, then we set up to understand what would be the evolution of coherences under the particular conditions that we show. What I am presenting here is, so we start in an initial state that is an excitonic state, but because of the interaction with the bath, this is not an eigenstate in which the system would relax. The system would relax in a different basis. So we're plotting here coherences in that thermal relaxation basis. So in that case, you do have certain coherences, a different zero value from that coherence. But the initial value is not so important. What is interesting here in the evolution of that coherences is that you have a first initial bang onto that coherence. So you have a particular frequency associated to this short time scale. And then, although you cannot see it in the distance, there are small oscillations superimposed in the evolution of these coherences that are here. When you do the Fourier transform of these coherences, then you are able to identify different frequencies associated to this short time scale and to this long time scale. In the short time scale, we are able to identify that the frequency of these oscillations match very well the energy differences between exciton states. In the long time scale, you see that the frequencies that you are identifying here match better the vibrational. Now, how do you distinguish them? Is because even though they are in close resonance, they are not banging on resonance. So you do have still room to distinguish slightly them. So then we speculate that what we are seeing in the evolution of these coherences is essentially in the short time, you see a coherent evolution associated to the electronic Hamiltonian, but this evolution is modulated by the bang that the vibration is giving. If we wanted to try to connect this to what is seen in the experiment, they do see these two time scales for oscillations, different kind of frequencies of oscillation. And this might relate to what they observe as vibrational coherence. But I will comment a little bit more when I present the last part of the work. What is interesting is that the two time scales that we identify here are comparable to the experimental time scales. So after this study, then I can now pose other two roles for these vibrations in this system. So as I said before, these vibrations promotes a transfer of energy that doesn't follow from up to down, but that actually follows whatever it is needed to spread across the complex, the excitation. And for comparison, I am presenting what you get when you have these vibrations in, or when you exclude them from your calculation. So when you exclude them, then you start with the energy in the subunit that is a higher energy, and it goes down in energy. When you don't have that, when you include your modes, then you have a more diverse transfer, which is actually dominated by the electronic coupling, even if the electronic coupling is small. You can see that better if you now compute two measures that in one, you are computing the population of the acceptors. The acceptors will be the final chromophores that receive the excitation. And you see that with and without the modes, you see a faster transfer. I have plotted this line here in the 1 picosecond because this is the relevant time scale in which you will transfer from one molecule to another molecule. And you see that in that time scale, you do have, of course, fast transfer with the modes. But most importantly, this quantity that I am plotting here is the variance in the position. So if you imagine that you can locate the excitation at each time in each chromophore, this one is telling you how spread is this excitation in the complex. And you see that the spreading of that excitation is larger when you have the modes than when you don't. But the most important result that this, well, not the most important, but one interesting result that we obtain from these calculations is the following. So we now see that these vibrational modes, which I am representing here, interacting with the two-state system, in particular with the excited state of that two-state system, are the ones responsible for this efficient behavior. So we set up to do the following calculation. We're going to compute a calculation in which we have no coherence whatsoever. So what we have is a non-Markovian incoherent transfer. So you have incoherent transfer, there is no Hamiltonian evolution at all. For that, what you do is you consider that these modes are very broadened, that the interaction to this phonon bath is so large that no coherent dynamics survive. And you compute your dynamics, the probability that it is transferred, the excitation is transferred from, I will call this the donor to the acceptor. Here I am mentioning the particular pigment or chromophore that I consider, but you can imagine just these two in which it satisfies what I have been saying before, that you have an energy gap that matches more or less the frequency of the vibration that is involved. And you compute that non-Markovian incoherent transfer. You observe that your population has these superimposed vibrations, which are essentially representing non-equilibrium behavior of your vibration that your population is able to capture because of this interaction that is relatively strong. Now, let's suppose that you still keep an incoherent interaction with this vibration and that you put some kind of correlations in your phonon bath, which can come, for instance, if your cutoff frequency is lower, so you have a slowly relaxing thermal background, or if you have some kind of solvent-induced spatial correlations in the system. But whatever it is, but it's a source of coherent dynamics. That means that if I now plot my coherences, I will have these well-defined oscillations. So this is the case. We compute in that case, and we see that now in that case, you do have an increase of population transfer. So I would like to stop for a moment here and let you think a little bit about this result, because what this is telling us is that in the presence of these vibrations, an interplay with a thermal background, let's imagine for now fully incoherent interactions with these vibrations. Only the effect of a thermal background that is able to support just a little bit of coherence is enough to improve transport. So for me, this is hinting that there might be a role for coherence, and coherence might indeed make the system more efficient, but you need to take into account not just this thermal background as it was taken so far, but you need to take into account these vibrations. If you want to think of the physics behind this, it's not so difficult. The physics behind this is the following. So when you have that vibration, what is making the vibration that is activating transport, what is making this transport to happen is this close resonance in their energies. Now, what this thermal background is doing is that now that is inducing some coherences that is slowly relaxing, as Francesca described, and it means that during this slowly relaxation, it induces coherent evolution, and that coherent evolution essentially keeps in sync the fluctuations that you have of the energy gaps. That means that the resonant condition between your excitonic state and the vibration that is close by is kept for longer time. And because of that, you have this improvement transfer. So it seems to me that these vibrations might be hinting to, yes, there is a role for coherent dynamics, and the role is indeed to increase efficiency. But if you don't take them into account, I don't have the calculation here, but maybe I can show, well, it's in the paper. But if you don't take those vibrations into account, and again, you compare two situations, let's suppose that you eliminate these vibrations, and you compare these two situations, a non-Markovian incoherent transfer, and then now you induce some coherence, most of the results that were at the beginning, let's say like a year ago, many people published papers inducing coherence. The result was that actually coherence in that case, and by coherence means coherent evolution of your superpositions, will actually result in a slow transfer. So in the absence of vibrations, the effects of that coherence, in this case due to this thermal background, was actually to slow down transfer. But in the presence of those vibrations, because of this matching of the resonance, but not just matching the resonance, but keeping in tune, that means keep the phase relation so that these fluctuations are correlated, then you have an improvement transport. Yeah?
So that means that basically if you were to lower the temperature, you would lower the efficiency. I mean, if you lower the temperature, you kill the bath, basically.
If you are a lower temperature?
Yeah.
Yeah, you kill the bath, exactly.
And so you basically go into the incoherent regime.
In this calculation, yeah. So you assume that you have no coherent interactions, etc. But I'll show now that actually discarding the coherent interactions here is not so good, because these modes, depending on the energy, might be not so broadened and actually might be sharp, and other effects can come into play. But what is more important is actually the temperature dependence. Once you have this mode, is very little, and the explanation is simple. Vibration is the one that is activating the transfer here. Thermal fluctuations are not activating so much transfer, so it is insensitive to temperature because of that. Because this vibration is high energy. If you had a vibration that is thermally activated, then the temperature dependency is different. But in this case, the temperature dependency is not so bad. And actually, it's what they see. There are no reported experiments, but I know that they've done experiments at different temperatures, and they don't see the difference much for these systems. And the reason is precisely because what is activating transport is a vibration that is not thermally activated. That is actually the matching of the energy gap with that vibration that is doing the trick. So I think there is a lot of room for interesting behavior in this direction. So what we are doing now in this direction now for this, so after doing this study, there are two directions that we are following. The first one is now I want to set up the task to unambiguously show that if you have these conditions and you have any source of coherent dynamics in this case, whether it is coming from a slowly relaxing bath or from actually quantum coherent interactions with this vibrational, any coherent contribution will help transport as long as you have a nearby resonance vibration. So this is one of the tasks we are set up now to do full quantum calculations with a molecule, the one that Francesca presented, just because we have more experiments in that molecule. So we can relate a little bit more to the experimental data in that case. But more as an example to show that at least in these conditions, coherent dynamics will increase transport. And that actually, as Francesca showed, you don't need to start in a particular superposition of states, but you actually can start in an excitonic state. There is another discussion whether that would be the right state, etc. But you can start in a variety of initial states and still coherent dynamics will arise and will imply that you have an improvement in transport or transport efficiency. But the second line of work that we decided then is now we want to understand in detail that this small unit, this dimer in which you have this coupling to these vibrations. I want a full quantum calculation of this, and what I would like to focus here is now not in the observables of the electronic degrees of freedom, but in the observables of that vibration. And what I would like to show is that in this case, when you have high energy vibrations, you have truly signatures of no classical behavior associated to these vibrations. A challenge would be how to measure them, how to witness them in an experiment. But at least for now, I just would like to pose the idea that that is possible. So in this case, again, so I remind you of our little basic quantum unit. And the energy scales that I'm interested, we've done calculations for many different parameters, but in particular this one because it's representative in many biological molecules. You have the case in which actually the lower energy scale or comparable to the bath energy scale is the electronic interaction. And you have your thermal energy scale is very low in comparison to the vibrational frequency, which is in resonance with this energy gap. And the basis of states that I decided to work here, simply because of the purpose of the problem, is a basis in which these X and Y represent the excitonic states of this dimer without interaction with the vibration or the phonon bath. And this 210 represent the phonon occupation of these vibrational modes. So for me, my system is going to be this extended dimer plus vibration, which we can call exciton vibration or vibronic exciton. You can call it in whichever way you decide. But the important point is that I have extended my electronic degrees of freedom to include a specific interaction with these vibrations. And I will consider then the effect of this thermal background. And the purpose here then is, instead of focusing, even though I'll show the calculation for the electronic coherence, I'm going to focus on observables of these vibrations. And my purpose is to identify non-classical behavior of these vibrations. Now, in order to do this, one have a few lessons from quantum optics. And quantum optics teaches that the state that is that is a quantum state closely related to a classical state is the coherent state of a bosonic field. So a coherent state is, for instance, is associated to the classical evolution of a harmonic oscillator. It's a state that essentially represents wave coherence, like we could understand any coherence in any wave. And it's a state whose coherences, different degrees of coherences, match, which means that it has a Poissonian distribution, which means that all the moments have the same. So this characterization of a classical, even though it's quantum, but it's a classical analog state, then allow us to think what would be non-classical. And there are several works in that direction, and recently in the last few years you have a very good characterization of when there is a deviation from that classical behavior. But the original works were from Glauber and Sudarshan, in which they do a peer representation of the coherent state, and you use the basis of coherent state to represent any state of any system. And then it turns out that when you set up for the task to know what is this P distribution of your coherent states, there are some systems that will have negativities in this that is supposed to be a probability. It's actually a quasi-probability because it's over-complete. But when you have negativities in a probability, then you know something weird is going on. And they identify these negativities as signatures of non-classical behavior, quantum behavior. So this is one approach. But there are other approaches that are directly related, simply because it's not easy to find this peer representation. So these other ways of identifying non-classical behavior have to do with hierarchy of expected values. Whether they are mean values, then the higher moments will give you fluctuations and so on. There are others that are hierarchies, not now in expected value of, for instance, number of particles, but are hierarchy of correlations. Like, for instance, if you want to characterize actually the coherent character of light, you need to calculate first order coherence, second order coherence, and so on. In this case, you are always quantifying how you are deviating from this classical wave behavior, classical coherence, so in which you actually have quantum coherence. In this, within this family of hierarchies, there is a very simple measure that one needs to be careful when one uses it. But if you find the right conditions to use it, then it's a simple measure that will give you an insight into the non-classical behavior that you might have in your system. And this is the Q-Mandel parameter, which essentially compares the variance of the number of phonons. In this case, it has to be a bosonic field. I mean, you can calculate this for fermions as well, but the interpretation is different. But in the case that you have a bosonic field, this Mandel parameter compares the variance of the number of the expectation, the variance of the number particle operator in comparison with the mean. So if you have that this parameter is 0, which will mean that the mean is equal to the variance, then what you have is your coherent state, and then you describe that as having a classical coherent state. When you compute this for different other kind of states, for instance, if you compute it for a focus state, which is a state that has a well-defined number of particles with completely random amplitude, or amplitude 0 in average, then you have that the Mandel parameter is -1, so it's less than 0. You can have your thermal state, and you compute in that case that is an incoherent state, and in that case you see that your Mandel parameter is larger than 0, so it's equal to the occupation number. But you have also these fancy states in which you have a focus state that's been displaced. So it's like a focus state that you give an amplitude. So it's like some kind of driving in this originally selected population of phonon states or bosonic states. And in that case, you do have the chance to still have non-classical behavior. So this is just to give you a flavor that this parameter, even though it's not decisive, yes, you have quantum and I'm showing your superposition, it does give an insight when you have classical or non-classical behavior. So we are using this parameter to quantify this non-classical behavior of the dynamics of the vibrational mode. And just a brief overview of the setup that we are using. So just to remind you, our system includes that vibration, and we are starting with an state that is an eigenstate of the excitonic system. The setup that we're using is this non-perturbating second-order cumulant expansion of the density matrix that is solved via a hierarchy of equations, as Francesca showed, originally proposed by Tanimura and now been changed, extended, studied, etc., by others, among those is Ishaki. And then I would like to show you the results here. So first, let me go back to population. So the first slide, the top graph just shows the coherent dynamics of system plus vibration, electronic plus vibration, so my extended system. And you see that you have population going to the other excited state, back and forth, back and forth. As you include these figures here represent inclusion of the coupling to the thermal background, of course you are going to damp those oscillations, but you still see, for instance, this is a coupling that is around 10 wave numbers, you still see these large rates of population transfer and small oscillations. The line here, the dotted line, represents when you have no modes. So when you have no modes, essentially you have no transfer, which is what I mentioned before. Now the figure here shows now the calculation for the parameters that are found in some of these systems. This line represents the dynamics when you have no modes, and now when you have modes, again, you see that this is essentially a vibration-enhanced transport. So again, just to remind that this vibration is activating and enhancing. It plays a fundamental, a significant role in the dynamics of the transfer. Now, these are the off-diagonal elements that represent superpositions of excitonic states having your vibrational mode in the ground state. And you see that now, obviously, because you have a state that is not an eigenstate of the full system plus vibration Hamiltonian, and then you have evolution of coherences. And you see that those coherences evolve as well. Here I am plotting actually the absolute value of this coherence rather than the real part of the imaginary part is the absolute value. And you see that this evolves with a particular frequency that is comparable to the frequency of population transfer, as it is expected, and a superimposed bit that is associated to the interaction between the electron and the vibration. What is interesting is as you increase the coupling to the thermal background, you still see, for intermediate coupling, you see, or for weak, relatively weak coupling, you see still this kind of population modulation of the coherence and the superimposed bit. But as soon as you increase the coupling very, very long, then you see that that coherence, at the beginning, you do have still that effect, which is essentially a similar effect of the effect that Francesca was describing. But the thermal background damps those population modulation of the coherences. You have the superimposed bit that comes from the vibration, but you do not have this population now modulating your coherences. And as you see, as you increase this coupling to the thermal background, any coherence here tends to be destroyed. This is not the case when you consider now the coherence or the non-classical behavior of the vibration. In that case, let me show you first, when you have no electronic coupling and no coupling to the environment, then you have, if you compute that Mandel parameter, you have that Mandel parameter goes from 0 to a particular positive value, which means that in the absence of electronic interaction, you have a classical. And this is what is happening. You have an electronic excited state that is exchanging with this harmonic oscillator. All you are doing is displacing your harmonic oscillator, but it's still a classical harmonic oscillator. What this figure is saying is as soon as you turn on the electronic interaction, that is, you have an excitonic component of your system, then that brings out the non-classical behavior of the vibration that you have. This is the Mandel parameter. Again, this is purely the classical dynamics. Now let's see what happens when you include the thermal background, which is going to damp your coherence or non-classical behavior. So and I'm going to focus on the average of that non-classical behavior in a particular time scale, which would be the time scale of population coherence, simply because that's the time scale in which I know my populations are going to modulate the excitonic coherences, and that's the time scale in which I can see whether there is a kind of effect associated to these quantum coherences. So if you see, for a system that is oscillating back and forth, you have the maximum negative value. And you have these green shaded areas are representing that I'm interested in the negative parts. What is interesting now is that if you increase the coupling to the bath, actually the system spends longer times with negative values. The maximum negative value is not as large as in this case, but the times that it remains with this kind of, in this non-classical states is longer. as you increase the thermal background, then you see that the non-classicality that you had at the beginning is washed out, but it evolves slowly because it is evolving with population evolution towards that. But in this time scale, which is the time scale between populations and coherences being coupled, then you don't have so much as effect. What is interesting is that it's not a monotonic decrease of that. Actually, if you compute these averages in this time scale, let me just go back to this. You see that what I'm plotting here is now an average of that negative quantity, and I'm just plotting the absolute value of that in that particular time scale. And you see that actually, as you turn on the interaction with that, with the thermal bath, you see that there is an optimal value for which you obtain this optimal non-classicality, and that is associated to an optimal transfer. This is not the regime in which these biological systems work, but I think it's an interesting result because it's telling you that this system is able to exploit something that is very closely related with quantum stochastic resonance. It's not classical stochastic resonance in the sense that the quantity that is optimized is a quantum not classical feature, which is this one. But it's still interested that in the biological regime that is around this case, for this dimer, you still have a large non-classicality of your vibration. And if you increase that non-classicality, that is accompanied by increase of population transfer, which is not, I'm not saying that one leads to the other one exactly, but they go together. It is not the same with the average of the excitonic coherence that I showed, because that one essentially is nearly dying. And with this, I would like just to finish. Just my last slide, and it's the dimer that I've described, and this small biological unit seems to be present in many systems many biological systems that transfer either excitation or electrons or protons. You can find it in the light harvesting. It seems to be responsible for the directionality of charge transfer in reaction centers. It is associated to the models that people have described for how we smell things. It is associated to a current discussion of proton transfer in hydrogen. So therefore, the direction that my research is taking is actually now to focus on the same kind of model, but now for the particularities of the system, in particular for this charge transfer in the reaction center. And with that, I would like to finish by thanking Edward, who is a PhD student working with me, who's done the majority of the calculations of the second part, and all the other people who I normally discuss and bother, and my funders. Thank you very much.